Good afternoon, good evening, good morning, good whatever time it is, wherever you are, and welcome to this Cloud Storage Technologies SNIA presentation. Just to make sure you're on the right presentation, this is Moving Genomics to the Cloud, Compute and Storage Considerations. And it is a live webcast as of September the 9th, 2021. So, without further ado, I'm going to introduce myself. I'm the chair for today. My name is Alex McDonald, and I'm an independent consultant. I'm the chair of the SNIA Cloud Technologies Initiative. And I'm joined today by three really good speakers. I've seen the slides, by the way, in advance, and they are terrific. If you like large numbers, by God, have we got large numbers for you. So we're joined today by Michael McManus, who's Director of Precision Medicine and Principal Engineer at Intel. Good morning, Michael. I think it's morning where you are.
Thank you.
Torben Kling-Petersen, who's a Principal Engineer at HP Storage BU. Good morning, Torben. Is it morning where you are?
No, it's actually evening, but that's okay.
Evening. That's exactly where I am too. And Christopher Davidson, who's the Applications and Performance Engineering Manager at HPE. Morning, Chris, or afternoon? He's on mute. Hello, Chris.
Getting off mute. It is morning with me.
It's morning, a muted morning. Welcome, guys, and thank you for helping us do this presentation today. Before we go any further, just let me give you a brief introduction to SNIA. We're 180 industry-leading organizations. We've got about 2,500 active contributing members and 50,000 IT end users and storage pros worldwide that participate in things like this at these presentations. You can learn more at snia.org/technical, and you can follow us on Twitter, the famous Twitter, at SNIA. Now, what does the CSTI do? Some time ago, we were just focusing in on cloud storage, but I think that we can all agree that cloud storage is much the order of the day these days. And what we now do is broaden our message out to cover the technologies that use cloud storage. As today, we're going to be talking about genomics, but we're going to be relating that back to the storage requirements that these new applications and technologies demand. So our goal is to educate both vendors and users on cloud storage and data services and orchestration, to do support and promotion, for instance, things like OpenStack or software-defined storage Kubernetes, to understand hyperscaler requirements, or dealing with cloud providers, especially the larger of them, and collaborate with other industry associations where appropriate. So that's what we do for a living. It's very much an educational role. This presentation, just to remind you, is a product of SNIA. We own the copyright to it, and there's also no warranties expressed or implied. In other words, use this at your own risk. If whatever you do causes whatever you're doing it to break into two parts, both of those parts are yours. That's the legal bit over and done with. Before we start, I'm going to cover the agenda in a second. I'd like to remind you that on the way out of this presentation, you can rate the presentation. You can rate it from 1, I wish I'd done something else, all the way up to 5, more please. We're hoping that we're going to get a good score today. And you can also ask questions of the presenters. So if you wish to pose a question during this session, please do so. If we don't get round to answering it, we will answer it in the blog, and I'll give the details of that blog as we end the presentation today. So please rate us as you leave, and also please feel free to ask questions should you wish to. So today's agenda, we're going to be talking about the trends in genomics and the need for data storage and management. We've always had that need, but genomics makes some terrific, put some terrific pressures on our storage environment. And we're going to cover that and the data management and storage considerations. And then we're going to talk a little about public science in practice. So our first speaker, I'm going to hand it over to Michael McManus, who's going to give us trends in genomics. Michael, over to you.
Thank you very much, Alex. Hello, everyone. My talk is going to have some big numbers in it that Alex referred to make a point. So let's get started. This slide here shows that there is a lot of genome sequencing already going on in the world. This is a map that I've been maintaining along with some colleagues that shows a lot of the human sequencing that's actually going on. This is whole genome sequencing that's happening around the world. And so you can see on the map where the numbers are. And on the left, there's some other programs that are actually happening. This Earth Biogenome Project actually is non-human. But by and large, this is all of the actual population sequencing that's happening. It doesn't include sequencing for cancer, for rare disease, and other forms of sequencing I can touch on later. So what's driving a lot of this sequencing? Well, uh, understanding our populations is being driven in part by trying to make better medicines and trying to make medicines more tailored for different populations. But another big factor is cost. And this is a chart that runs from, it's from the NIH. And you can see the original, $10 million in 2007 is now $689. And this drop is a lot of the impetus behind the sequencing. So there are lots of sequencers out there. Traditionally, Illumina has been the lead. That's this left panel here in yellow. But of course, Thermo Fisher over here in orange has been around for a long time too, and has acquired the original Applied Biosciences sequencers. And so those have been around also a long time. And of course, here we have Beijing Genomics sequencers. And then we have PacBio, which are long read sequencers, as an Oxford Nanopore. So you're seeing not only a drop in the cost of sequencing, but you're also seeing a proliferation of the number of sequencing instruments that are available on the market. So let's just talk very briefly about human, genomics. And I'm not going to explain all these steps, but at a very high level, you're taking blood, for example, from a patient, or tissue, and you're extracting DNA, actual real DNA from that tissue or from that blood, and that's what's represented by these little test tubes here. And you're placing that DNA in what's called a flow cell, which goes on the sequencer. And this is kind of an Illumina example. And then the sequencer is going to convert this physical DNA into a digital representation. And these are the file names, which, in bioinformatics, everything's an acronym or complicated, but just think of this as a digital representation of the real DNA. And it's this digital representation which then is used on computers. It's called secondary analysis to go from, this raw digital representation to a list of variations that are different from a standard reference genome we have for humans. And it's these differences for a particular patient that are indicative of perhaps some medical issue or some inheritance issue or any traits that they got from one parent or another. So the idea is, you figure out, at least in the medical context, if someone's sick, this list of variations can be the clues to help find out what the right treatment might be for that particular patient. And the rest of these are all sub steps, computational steps that I won't bore you with. So I want to talk about COVID for a sec, because another form of sequencing that's happened, and it's not necessarily purely cancer, it's not inherited disease or rare disease. This is sequencing around COVID. And so, just a little timeline here. As we all know, in January, of 2020, we saw the first, genome from China for the SARS-CoV-2, for the coronavirus. And then what you'll see here is that, I'll just pick a few points. These numbers are on this yellow line or listed here. You see in Australia, they had 1,300 cases and 80% got sequenced. And they identified that they were coming from basically uh, outsiders, if you will, outside of Australia. And then the UK did a bunch of sequencing. They sequenced about 6% of the cases at the time. That was more than anyone else. So number 6 is 4 is right here for the UK. And then, we saw this big surge in South Africa, this B1351 variant. And then in Brazil, they were sequencing about 40% of the genomes, and they found that it was P1, which is now called the gamma variant. And so this activity has been going on, and of course the U.S. started sequencing from the CDC, And so this was another unintended, at least or unplanned source of sequencing that happened. And a lot of this data, um, when the sequencing happened, ended up noting, the time and location of where the sample was collected. And there's this great website called nextstrain.org. And that website, I'll show you here. So this is an example from, say, September of 2020 to February of this year. And these are colored by the current variants. So you've got the beta, which was the one that took over England early, gamma, which is the one that's in Brazil. And you can see these little pie charts here are colored by various, the occurrence of these various variants. And so in this little window here, you see mostly the beta variant in dark blue and the gamma variant, which was largely in Brazil and South America. If we step forward in time, so we're going to go from September to this window to February to May. And now what you start to see, which is really interesting, is a rise. You see the very beginning of the rise of the Delta variant, which is this color here, and you'll see it's taking its chunk of the pie in a lot of these diagrams here for each of the countries. So all of this sequencing and from patients and of the virus itself, has been contributed to this kind of database to track what's going on. And if we step forward one more to August of this year, so just last month, you will see the predominance of the Delta variant here, and you'll see how this green is like everywhere. So all of the sequencing that was done has enabled people to, or at least the community, to track where these viruses are coming from and how they're growing and proliferating. And this last sort of overview slide here just shows the whole timeline from December to August. Anyways, so you can see there's
Michael, just a quick question from
Sure.
me, because I think I'm seeing something here. These are presumably family trees of viruses.
They are
Yeah.
clades, CLADE,
Clades, yeah.
which are effectively families, and they show you how they've branch out and how you see them start proliferating. And so you can see there's like a dendrogram, if you will, if you know what that is, that generates these.
Okay. And how large is the genome?
Yeah, good
I'm
question.
thinking of the kind of storage that we have to commit to this kind of stuff. I mean,
Well,
people are presumably sampling, taking the genome, storing it somewhere. I'm just trying to relate this back to the storage. How much storage is this? Is this just this particular set of examples consuming? Do we have any idea?
I don't have that number, but the average human genome, if you don't have any compression, just a big number, is about 350 gigabytes. And then on top you have the viral genome, which is not as large, but still it adds up.
Okay, thank you.
Yeah, sure. So let's just talk about, since that was a good segue, let's talk about data volume. So let's take an Illumina NovaSeq here, and depending on which kind of flow cell you use, we'll just use one that's kind of medium. You can do 360 whole genomes, that's what WGS means, per month. And so if we know that we retain about roughly 350 gigabytes, 0.35 terabytes per whole genome, it means that this instrument's generating about 126 terabytes a month. And then the question is, I want to move that data from where it's generated to someplace where it will be analyzed. So if you remember in my earlier diagram, we had the sequencer going from from real to digital, and then we took that digital representation and processed it. Well, that processing happens typically in a data center or on the say the public cloud. So now we're faced with moving this data. So you can move this many terabytes a day per sequencer, or you can stream it, which is something Illumina did a long time ago in their incarnation of base space that it runs on Amazon. It's now been superseded with a new version, but you could stream it, in terms of all day as it's generated, and then process it once it's done. So, but still, one sequencer, and imagine hundreds and hundreds of these sequencers around the world generating this kind of data and moving it, it adds up quickly. But the generation of data is one thing, and then the actual what's retained is another thing. So this slide is an older document, which it's I think like 4 years old. They were discussing who are the biggest generators and retainers of data, and astronomy won hands down for the largest generator of data. But the astronomers have really good ways of doing data reduction, so they don't retain as much after they've generated it. But still, this is a log scale, not a linear scale. And so this data is probably a little out of date, but you can see YouTube is forecast to have 2 exabytes of data, so 2,000 petabytes, so 2 exabytes. Genomics is for data retention is slated to have 40 exabytes of data, and this is before any of us realized about all the sequencing related to, say, COVID. And some of the other sequencing that's been happening. There's a lot of new kinds of sequencing, single cell sequencing, spatial transcriptomics. I mean, there's a lot of things happening that weren't anticipated when this document was written. So I think this number, if it were reevaluated, will be even larger. So let's just talk about, like, what if we wanted to just sequence the entire planet? And, if I wanted to, like, take everybody, so we got 7.8 billion people roughly this year so far. And this is just a pie chart that shows the biggest, country actually is China. India is in here with Asia, and so Asia is the actual largest chunk, even without China. And then these are millions of, or millions or of people. In this case, 1.4 billion. India is like 1.3 billion out of here. And so if we wanted to sequence everyone, this is the kind of pie of population that we're looking at. So not insignificant, but if I convert this pie actually using some basic sizing metrics, so let's just use 350 gigabytes per sequence. And what happens is you can convert this to actual, in this case, exabytes, which are these numbers. So this is thousands of exabytes. And if you know what that is, that's an even bigger number. So zettabytes. So basically, you're looking at 2,700 exabytes to get all about 7.8 billion people on Earth. Now, if you do this over 5 years, for example, then this number will grow over time. And so this is just kind of a really quick thought experiment here, and you can read some of this data comes from these references down below. So basically, now what is this number? 2,743 exabytes. What is it? How can we visualize that number? So I like to do this kind of experiment. I used a metric version here, but basically, I'm going to use a 10 euro note as our basic measure here, and I'm going to assume 1 byte of all of those exabytes is about the same thickness as a 10 euro note, which is just over a tenth of a millimeter, 0.15 millimeters. And then I'm going to imagine a stack of these things to total up to the 2,743 exabytes. And doing that, of course, I'm ignoring physics and relativity here, but this would be a 412 trillion kilometer stack, which is about 43.5 light years. So a star that's in that range is this 58 Eridani. So we're talking about an enormous amount of data, not including, like I said, sequencing for cancer, sequencing for rare disease, single cell sequencing, spatial transcriptomics, and other related kinds of sequencing that's happening. These are all contributing to a much more massive storage demand for all of this data. And I think that might be my last slide.
Thank you, Michael. I'd just like to make a couple of observations. Firstly, you may be aware that we measure area in this country, in the UK, in Wales is something is the size of Wales. Nobody actually knows what the size of Wales is, but that's how we measure things. Texas is about 35 whales is. This is taking the whole thing. Do you remember we used to do pizza boxes?
Ah, yes.
Yeah, I'm just wondering how many pizza boxes would we actually reach Andromeda with pizza boxes? Anyway.
Well, I mean, yeah, I mean, a pizza box, what about it? And a little over an inch. So it's about 2.5 centimeters. So you could, if a byte was 2.5 centimeters, it would be way larger than this, right?
Yeah, the numbers are pretty scary, but I do understand the point you're trying to make. We're actually, growing large quantities of data here, and really, I suspect, haven't thought about the management of them. I've had a couple of questions in.
Yep.
One of them I think is for you, but I'll leave the second one, I think, till a bit later. The first one is, what is the size of a sequence? How many sequences are we looking at? I suspect you did answer that in the subsequent slide, but could you just
say it again? How many sequencers are there?
Yeah. What's the size of a sequence?
Oh, the size of a sequence. So the sequence is represented by letters, but it's DNA is a large molecule, a double-stranded molecule. It has 6 billion bases, which are the components of DNA. 3 billion base pairs, so that they're paired across each other. And so the size of the sequence itself, if you want to represent it with 1 byte for the actual letter and 1 byte for, say, a quality score, then it's really about 6 billion, right? If you want to think about it that way. And then in sequencing, we use coverage as a way. Coverage is a sort of a resolution. You run it many times, and you do that to distinguish between an error in the sequencer and a true variation in the actual DNA that you're looking for. And so if you multiply 30 times, say, 6, you get 180 billion. So you can start seeing how things add up, and then you process that data and you transform it, and you end up accumulating more and more data that totals up to this roughly 350 gigabytes.
Okay. I think we'll answer the question about how these things are stored and processed, and when we get a bit further through the presentation. So let's move over to the storage challenge, because I think we've seen the scale of the thing. I mean, it doesn't matter whether you actually think we're going to sequence everybody in the world or not. It's just the fact is that even sequencing a fraction of that number is a scarily large amount of storage that we have to deal with. So I'm going to pass it over now to Torben Kling-Petersen, who's going to talk us through the storage challenge. Torben, over to you.
Thank you. So I will actually try to be a little bit more on the technical side and not necessarily hit these super high numbers, because it's a challenge for us to build systems that can cope with these over time, because of not just the size of the system from a physical point of view, the power requirements and so on and so forth, but managing these namespaces when they start growing to an exabyte in scale is actually a monumental task. Everything breaks the scale. Now, we were talking about 2.7 million, 2,700 exabytes, and an exabyte, as we already mentioned, is fairly large. But there is no file system, single namespace at least, on the planet yet. There is in that range. The largest ones currently out there is a few hundred petabytes. We are actually building one at the moment that is hitting about 700 petabytes in a few months, and those are exactly breaking the records of what exists today. Now, these kind of file systems have been around for a long time. The HPC file systems previously used for much of the modeling, the supercomputers you've heard of with Oak Ridge, Lawrence Livermore, CSTI, the K-computer in Japan, and so on and so forth, all have been around trying to simulate a reality, whether it's a existing reality or a potential future reality, whether it's earthquakes, whether it's atomic explosions or what have you. It's all about simulation. And then you can always look at the results afterward and compare with the real thing, like the weather services are doing. Machine learning is, the total difference of that is about looking at what you have and trying to figure out what you don't know about something that may or may not be entirely unknown into the future. And those two disciplines have been very different for a long time. So if you look at the HPC systems, they have mainly been about writing a lot of data. It's been about large files, doing checkpoints and what have you on these supercomputers, whereas machine learning is about trying to feed GPUs with specific data that's transferred into numeric values. So a lot of small files, a lot of random access, and a lot of I/O going on that isn't necessarily putting a strain on the bandwidth, but putting strains on the capabilities. And if you look in historically, that's been different types of systems. So for instance, on the HPC storage side, you've had the cluster systems that I've been working with for the last 10 years. You've got IBM's GPFS systems, you've got DDN's equivalents to those, and so on and so forth. Whereas AI storage have more been around small NFS-based solutions, either flash-based or in some cases disk-based. But what's happening now is that the AI storage is becoming data intensive, and the HPC compute system is becoming I/O intensive because we are seeing a combination of these two technologies kind of merging into the future. So any and all supercomputers today, or at least most of them, even the exascale computers being built as we speak, have a significant amount of GPUs, but they also have a large number of CPUs that still needs to be fed with these things, and that's where the new generation of storage comes in. So if we just look at traditional, you had your compute system on the left, you had a fairly fat pipe talking to some parallel file system. You usually had an NFS component as well for home directories and smaller things, and you backed it all up to a tape archive. That's how we did systems 20 years ago. That's how we did systems 10 years ago. That's how we did systems just a few years ago. Now, moving this to the cloud is fairly simple because you're doing exactly the same in the cloud. You're just changing the backend to be a more cloud-based storage, but the front end still looks fairly much the same. And we have systems today in Microsoft Azure, for instance, that is exactly like this. You have a mainframe computer is connected with a high-speed interconnect to the storage, and you have access through other means, and you feed the HPC storage from blob storage at the outer edge. And this does work well. It is simple, it's straightforward, it is manageable. Unfortunately, the new requirements that we're seeing on storage, more GPUs, FPGAs, all these kind of different CPU types that has multiple cores, or in some cases super cores, and you have all these attempts to build GPU clusters that starts ranging into the exaflop range and so on and so forth, or the petaflop range. That requires something a little bit new. So the new world we are looking at still has your supercomputer on the left. Now that is becoming larger, it has more components, it has more GPUs, it has more cores and what have you, but it's still fairly much the same solution. But the storage part of this is now that you can start adding storage already in the compute side. Now we've been able to do that for a long time with no local storage. The manufacturing and automotive industry have done this for a long time with local disk drives because some applications really like a fast local temp space and so on and so forth. What's changed is we now have local storage that operates at memory speeds, like the pleated cross-pointer Optane, as Intel call it on the market, which is what's called a storage class memory. It acts like memory, but it actually retains the data like storage would, even if you power it off. Now, these are still node local. It means that you don't really share data between the different nodes without a lot of overhead. But there is also some advances here where you have a new type of file system called DAOS. And DAOS stands for Distributed Asynchronous Object Storage for short. And it's essentially an open source project that is allowing you to create a file system over these storage class memories in order to have an extremely fast and extremely I/O intensive storage that's actually shared over the supercomputer. This is obviously something that requires a good bandwidth in the network and so on and so forth, but we have that in most systems today. The problem with this is it's expensive. It is a little bit hard to maintain and support, but most importantly, you can't scale it to any size. It's limited to a certain capacity point that is larger than the primary memory computer, but way less than the actual storage needs of today. So now we add the traditional parallel file system that has changed to accommodate new types of storage. We still have HDD-based components because the cost difference per terabyte between an SSD and a spinning drive is still about a 7x per terabyte, which is a significant amount of money in the big picture. Now that still allows us to put some latest generation SSDs or NVMe's into the storage solution in order to keep that high throughput, high performance, high IOPS level working. But when you put all of these things together in a single namespace, you create a new problem. You create a problem where you need to have data services to maintain the different levels of the data in a usable form. And I'll talk more about that on the next slide. So the storage has now become very much more complex. It has more moving parts. Those moving parts are operating at very different speeds and very different mechanisms and have a very different life cycle as well. So a lot of challenge. You tie everything together with an RDMA-based network, so you can do offload from the CPU and don't have to actually copy things through the CPU using essentially one of these technologies. Now, there's still traditional TCP technologies out there that does have a little bit of overhead, but that's more for systems that need a special form of compatibility with very predictable rates rather than what RDMA can do. But you need more. You can't just have a supercomputer. You can't just have the storage, even though we're talking about 700 petabyte in some of these solutions out there. If we start getting into the multi-exabyte range, we also need cheaper storage. We need storage that can deliver the capacity at a much cheaper cost, but still be accessible. And that brings in a data management component. And whether we store this on what we call serial storage, which is essentially it's large storage devices, you can power off when that data is not needed, and you can power them back on, and you have enough erasure coding to still retain all that data should a drive or two fail. But you also have traditional tape. Yes, I know tape is dead. I've been saying that for 8 years, and I've been wrong for the last 8 years because tape is still the cheapest medium you can store data on. And a tape will last you for 5 to 10 years. So it is still something that you see around. It's still something that's been installed in greenfields today. So it is a valid component. You also have the traditional cloud storage that has its quirks and its benefits. But what's also been more and more common nowadays is using an object store like a data lake based on Ceph or Scality or something like that, that gives you a performance bracket in the between these three others, so where you can access data really fast, but you can also do it in fairly high scale, and you can share it in the other direction as well. I'll talk a little bit more about that as well. And again, what's now changed is that the fabric end to end now tend to be the same. So we're not changing fabric to a fiber channel somewhere. We're actually using the same infrastructure to connect from a compute all the way down to tape, which is kind of the big change. However, this is still very traditional. So what we're trying to do now with these systems is essentially create a single virtual namespace over everything. That means that you have a system that not only owns and can control all your data, it also knows where all the data is. It knows which version it is. It knows if it's a copy or a prior version of a file. It knows if it's HSM from the storage down to the secondary or tertiary targets. So you're creating a new environment where you can actually have a single view on billions of files at the same time. All of this is technology we can do today when we do this in most sites on premise. Now, moving this into the cloud has some challenges, which I'll talk about in a few slides. But I want to talk about these two components that we have in the system first. Data services, for instance. And the concept here is that flash is expensive, but you really want to use it as much as possible because that's where your performance is. That's where your IOPS are. And that's how you get most of the system out. However, you also need to make sure that that data doesn't fill up and you have to move that data at the wrong time when you're actually trying to utilize a lot of that space or capacity or performance for other purposes. So we need policy-based data migration. Essentially, we need systems that can allow you to monitor automatically a flash tier and says, okay, this flash tier is now hitting 85% full. Let's kick off a policy that looks for any file larger than 20 gigabytes that hasn't been updated in 48 hours and move that off to a storage, to a disk-based components. And that is exactly what data movement does today. You can also use these movements to purge files that isn't needed. For instance, every 24 hours you run a policy that says, find any temp file that hasn't been accessed in 6 hours and delete it. And that way you can always keep your flash component pristine. Now, the last part of this is what we call workload manager directives. And that is essentially that if you run a large job, it could be a genomic search, it could be whatever, it doesn't really matter in this case. You need to be able to launch that job into a workload manager like Slurm or PBS Pro. And that workload has, or that work order has, a prolog script that says, before you launch this job onto 2000 cores, make sure that this list of files are stored in Flash so I can access it quickly. And that means it tells the system, move the data from disk or from backends to the Flash tier. And once it's done, you tell the resource manager, you can now run the job. It runs the job, it accesses the files really quickly on the Flash, can execute it really fast, and you get the most out of your compute system. And once the job is done, you have a postscript that tells, move this data to this repository on disk or on tape or whatever, in order to save the space on the Flash tier. Now, all of this requires a search facility, and you can always search on a file system, but whether it's a large namespace on Lustre, Spectrum Scale, or any other of the parallel file systems out there, a large file system-wide search takes time. It takes resources. So you need to have a copy of that file system in a much faster searchable medium that you can query parallel to the actual parallel file system. That's an oxymoron. Okay, the parallel to it, in order to get good performance. And that's also something that's in these data services today, an externalized version that is essentially up to date, but you can search it much more granularly and much faster than you could do in a search onto a file system. And that allows you to have control over the data in a much wider sense. Now that brings us to the second part of this, which is the data management, where you also need to have the same type of policies moving data that's not used off the disk tier or the Flash tier for that matter, onto a cheaper medium. That could be anything from a hot archive on disk to a cloud ice layer somewhere where it's essentially stored for posterity, but it will take time to get it back. And now you need to have policies based not just on size or age, but also on the project type, classification, ownerships, legal requirements, and so on and so forth. So the requirements of metadata in this system increases. It also needs to be able to manage multiple different front ends, because you may have a Lustre file system, you may have a Spectrum Scale file system, you may have a DAOS system, and all of these need to be managed by the same entity in order to know where files are and be able to place them where they should do. Now, this also allows you to actually move files between these front ends. So you could move, using these kind of tools, the data from a DAOS to a Lustre, or from Spectrum Scale to DAOS, or in any other form, depending on what you have in your data center. And that is really the big change in how we manage data. It's not just about archiving, doing HSM, or doing XML, or doing backups. It's about actually managing this data in all four directions, left, right, up, and down. And that also means that you can have tiers that are really optimized for the purpose that they are built for, not just because you need a certain performance or capacity. Now, all of these functions exist today, but how do you move this to the cloud? And why do you move it to the cloud? Well, benefits are obvious. It's cheap in the cloud. You don't have to maintain it. You don't have to support it. You don't have to own it and pay things up front. You don't need to have your own data center. There's a lot of value in that, and you can pay as you grow. You can reconfigure your compute and storage whenever you want, and it's really a much more flexible tool. Well, there are some drawbacks as well, as you probably already imagine. One of them is that we moved the bottleneck. So instead of having to move data to and from the compute or to and from the ingest sites where the data is actually created, whether that is sequences or something else, you now have to move that data to the cloud. And that pipe is not necessarily predictable, and more importantly, it may not be as fast as we need it to over time, and you add a lot of cost. Then you have the problem of multi-tenancy of these systems, security, legality, what can be stored where. I mean, in Europe, we have a major issue at the moment with Brexit because we used to share a lot of data with the Brits. Now we can't do that anymore because they're not part of EU anymore and they have their own rules and regulations. So there's a lot of legal constraint that also needs to be accessed here. In compute, you often use containers in order to separate things, but data I/O in a container is really not a good idea. It is something that has a lot of overhead. It really slows down things, and it kind of negates the whole idea of having a storage in the cloud or storage close to the compute. And then you come down to the last question: how do you actually pay for things? It's not like you can just count clock cycles on a CPU when you do computations and you pay for those clock cycles. If you're storing a lot of data in the systems, are you paying for the capacity? Are you paying for the performance or a combination thereof, and so on and so forth? So it's much harder to get a equality in who pays for what. And then we have the benefits, which now turns into a detriment in storage. Simple reconfiguration. Well, it's easy to reconfigure, reboot a node with a new OS with a new set of applications. Now, if you do the same on storage, you have to copy the data off first to a secondary medium. And once you reconfigure it, you need to copy the data back on again. And that is also a challenge that we haven't really solved yet to the extent that we need, especially not where we have these huge systems, these exabytes of data, and these challenges that we are seeing being more and more frequent. So it is not a clear-cut, easy decision to move everything to the cloud. There are some challenges, and we can't solve them this problem, but we are essentially at a crossroads now where we need to figure out how to solve some of these problems. And I think that is my last slide. So
thank you, Tom. Just before we move on to Chris's part, remember to come off mute, by the way, Chris. I just want to thank you for two things. First of all, for mentioning Brexit. You don't know how much that pleases me. And secondly, are we, as an industry, going to end up with metadata madness? I mean, we've already got the issue. Well, there's a less of an issue. We've standardized on genomic data, and most genomic data conforms to certain standards. But we don't seem to have this metadata pinned down, or the way of managing the data, or even file systems. You're talking about single namespace, but that presumably is multiple single namespaces across multiple projects. I mean, how do we get around this problem of the proliferation of ways of managing the metadata?
So that's something that still needs to be solved when it comes to genomics and other things. There are attempts to try to do that, but if you compare with other industries, such as the weather service industry that has a humongous amount of files, they have very tight constraints on how these files are being packed, stored, and shared. They've actually come up with a solution where everybody uses the same type of metadata, the same type of embedded information, so you can always figure out where a file is coming from and where it's going and what it's been used for. Now, I've not seen that in the genomics yet. There are some efforts in Europe between Sanger Institute and EBI to come up with a common framework for how to do these things, but To date, I haven't really seen a global effort to try to standardize so that everything is being handled in the same way. And it is a challenge, I agree.
Okay. Yeah, it's just one that struck me when we were going through it. We really don't have any standards in this area, something perhaps we ought to be looking at. Anyway, so what's the next step? I'm going to hand over to Chris. Chris, away you go.
Hey, great. Hello, everybody. So far we've looked at trends in genomics, explored some traditional and newer architectures, and discussed kind of the positives and negatives of cloud computing. So we haven't really looked at what is driving the cloud initiatives today versus the need of tomorrow, right? Where do we get to that point where genomics overtakes astronomy data? And have those initiatives really work? Well, a lot of what's driving that is what we're going to call public science or open science. And unfortunately, yes, you're probably tired of hearing about COVID, but we're going back to the COVID example. So the HPC computing consortium built around COVID-19 is an example of that. And that brought together basically members from government, industry, and academia. Basically, this is in the States, early 2020, March of 2020, though it's expanded. And that ultimately allowed a number of researchers, PIs, access to some of the most powerful computing systems in the world. The consortium is actually still active today and has overseen some 100 projects. And let's say about half of those, or well over half of those, have incorporated or incorporate, since it's still active, genomic data in some way, shape, or form. So some quick facts and tidbits of info here. Consortium started relatively small, right? HPE was one of the original 13 members, of which 5 were only from industry, right? The rest was from academia and some from government. So even then, this initiative was still one of the largest private and public collaborations that we'd ever seen in the States between kind of industry, government, and academia. So even at 13 members, that kind of says something, right? It speaks to a trend that's starting to emerge, right? Basically, as the focus on COVID grew, so did the consortium. And the beginning of the year, consortium had 43. This year, the consortium had 43 members, and it expanded internationally and had a mix of projects submitted from, again, academia, private, right? So commercial entities, research entities, and even medical organizations. Kind of ran the gamut. So if over half the projects relied on genomic data, how did these groups gain access to the data in a safe and efficient manner, right? And my next section will kind of go into maybe focusing on the middle bottom of the screen, right? In May of 2020, there's the word Europe, right? Price and UKRI joined the consortium. This was on the coattails ultimately of the European Commission announcing basically an open science program and a data portal program, right? And this open science initiative is a really complex process ultimately, right? So looking behind the scenes, this actually aggregates data from both human and viral samples, along with a slew of other data, and consists of kind of 3 resources, right? There's the one SARS-CoV-2 data hubs, organizes the flow of outbreak and sequence data. There's other resources which are lined up with more EMBL, EBI, PDB databases, protein data bank databases, and other public databases. And then the third, which I find the most interesting, is a Federated European Genome Phenome Archive, which provides secure, controlled access to sharing that data, or sharing human data ultimately, right? So this is the one that's probably the most stringent, right? And all 3 of these are pooled together to create this data portal, right? And create a data platform. So that's what I've highlighted on the left, just a quick screen grab. On the right, so this is separate pages, right? Links are at the bottom of the screen if you'd like to visit them. On the right, we talk about access to the data being important, but what you use or how you use that data is also important. And this isn't meant to be an ad for Mulsi, but I find it quite interesting. There's a number of organizations that are built around this now, or built around these data portals where it's not just the data, but it's how you use it. It's ultimately best practices for building applications, using applications, and creating a portal for Q&A as well, right? So if you're a researcher on one half of the world, how do you find out how to best run VASP, right? Or how do you best find out to run Velvet, right? Depending on what you are researching, right? So kind of a novel approach, taking it one step further, right? Ultimately, portability, reliability, and repeatability being the main focus, and guaranteeing good quality science. But you know, as Torben mentioned in his portion of the discussion, right? These initiatives are great. They can be highly effective and efficient, right? You're able to just put data into the cloud and not think about it, but they're still less than perfect. Even with the COVID-19 data portal and the money and funds behind it, the way to get the data is still off a basic FTP, right? It can be quite slow. I've even tried to pull some of the data myself. Wasn't exactly successful, but again, I was trying to do different things in the background. Anyway, moving into the summary and kind of in review, it's maybe important to point out that the data for genomics is growing at an exponential rate, right? We're trying to keep pace with that. You know, growing more than YouTube, going to be more genomic data than there are cat videos in the world, which is unfortunate. But there's ways around this, right? We can work smarter, not harder. Torben spoke to this, right? You can't just save everything into Flash and just forget about it. You can't just toss it up into the cloud and expect to get it back quickly. And the other part of this is realizing that compute is really a small part of the problem, right? It's the data management and data storage that are of utmost importance, right? And then looking at public and private cloud, right? There is a difference between public cloud and private cloud. What are the differences? How do we manage those differences? How do we port data from one area to another? How do we replicate even from one organization? If I have another office in let's say San Diego and another office in Zurich, right? Or there's a research lab rather. How do I make sure that I replicate that data? Are there means to do this? And last but not least, you know, cloud provides a number of challenges, but nothing is impossible. You know, we're developing new ways of transporting the data, of saving the data, of utilizing the data, and yeah, kind of an interesting field, right? And Michael, I think that's the end of mine. So back to you.
Thank you. No, I'll pick up from here. Don't worry. Just let me. I want to point out there've been several questions and we haven't answered some of them. So we've got a couple of minutes. I'm going to throw out some questions here. Some of them are to do with compression, and I got a sneaking suspicion we could get rattled really easy to do compression and deduplication of data. But there was an interesting question about I/O patterns. You know, when you're dealing with this 350 gig object or whatever it's, but it's a file or something like that. What kind of I/O do you expect to see in it? Sequential? It's obviously, you know, read sequentially or sent sequentially from the sequencer, but what kind of I/O are you actually doing on it when you're processing it? Michael, Torben?
Yeah, so Michael here. So I mean, the 350 gigs is a composite of several files. The typical starting point is something called a BAM file. It's a compressed version of the raw data. That BAM file is about 100 gigs. And you can have many of these on disk, and this kind of processing is done on an HPC system, but it's really high throughput computing. There's no MPI. So basically you're reading a bunch of these 100 gig files off of disk into the CPU, into memory, and then you are processing them. And so having high I/O, like parallel I/O, is really important for that particular activity. And then this is a multi-step pipeline. There are many different steps in it. It's a lot of Java, not so much C++ or C. And so you have a lot of I/O patterns. You have some single-threaded pieces of the pipeline, and you have some multi-threaded. It's a complex, right? It's hard to give precise answers.
Yeah,
But the compression question, I mean, there are commercial solutions, and there's other compression methods. There's a method called CRAM, C-R-A-M, which supposedly can get to between 30 and 60% of the original size. Petagene, which is a commercial solution, claims to be able to get as high as 87% compression. And so depending on what you want to pay for and how your systems are set up, you can get better compression. My numbers were meant to be a maximal impact, not necessarily. I was trying to make a point, but yes, you can compress. And this question about 1% different, I mean, the challenge with that is that the differences are complex. They're not just single letter changes. There could be large structural changes. You can't actually represent them as just this 1% change. It's much more complicated than just the difference in genomes between people would make it appear.
Thank you. We'll reiterate that when we answer the questions on the blog. The other thing I'd like to point out is somebody asked about why don't you use something other than tape? Well, the answer is do a quick search on DNA storage.
Yeah.
People actually proposing storing information on DNA, which I presume you all understand that to actually read it, we have to sequence it. Hey, how about a bit of circularity before breakfast? Anyway, thank you for viewing this webcast. I'd like to thank everybody for coming along today. Remember, we will answer your questions. We're going to post the Q&A on SNIA Cloud. As part of this webcast, you'll find the slides are there as an attachment on the Bright Talk session, so you can pick the slides up. Remember the thing about the copyright on the slides and also the no liability. But we'd like you, as you leave today, to rate this webcast, provide us with some feedback. Again, a copy of the webcast and the slides, should you miss it, from this Bright Talk, you'll find on our educational library. We're going to post the Q&A. You can follow us on Twitter at SNIA Cloud. I'd just like to thank Michael, Tobin, and Christopher today for coming along and giving a really good, really interesting presentation and tying back in these big numbers back into the kind of storage requirements, and also not just direct storage, but the stuff that sits on top of it, how we're going to manage it with metadata, how we're going to build file systems out of it, the kind of technologies we're going to need to be able to process this stuff efficiently and effectively. But anyway, thank you very much indeed, gentlemen. Thank you to the audience, and we look forward to speaking to you again. Take care.
Thank you.
Thanks.